Well, kind of. It's not showing very well. Get it over there any better than that. Showing me most of my green screen. That's not good. Before we begin, are there any questions? I'm having trouble with my Zoom, so my background's not going to be very good. All right, today is July 15th, and let me share my screen here. So we should be starting chapter 2. I think we're there. Let me remind you, you have a practice quiz this Wednesday, practice quiz 00. If you get all the questions correct, you'll get an extra credit point. So each question, if you get it correct, it's something like 0.1 point. And you should take it because, first of all, it will make sure that you can work the lockdown browser. And then it will get you familiar with my multiple choice quizzes. So you will hopefully do better on a multiple choice quiz. Any question about any of that?
Criteria for that over chapter 1?
This is going to go over chapter 1 and obviously what we covered in chapter 10 and labs 0 and 1, as well as the syllabus. So the questions will come from all of those sources. And if you notice, I always give you what the practice quiz will be covering in the syllabus. Anyone got a question about that? All right, there is 2 labs we'll discuss today, and I need to introduce the infectious disease project. This project is starting today, so if you send in a request starting today, I will take them in the order of the request. So the first person who asks for, it's something that's very specific, pneumonia caused by an E. coli infection, you will get that because you're the first. The second person, I'll tell them they have to choose a second choice. And if you're early in your selecting an option for this project, you can give me just one choice. But if you're late, like after this week, you should probably give me at least 2 choices because a lot of the choices will be taken. It must be an infectious disease. It must be cleared by me. Everyone's going to have a different project. I do break up some projects like HIV. I break into HIV 1 and HIV 2. The diseases are slightly different. Tuberculosis, another very commonly requested topic. I break into late tuberculosis and early tuberculosis, meaning you have different symptoms if you're in late stage disease or early stage disease. And so write your project on one or the other. There's a few other projects I break up. I don't remember what they are. I guess malaria would be another one. I break it up by the specific species causing the malaria disease. The disease is a little bit different depending on what species you choose. Any question about any of that? All right. So let's begin with
Question real quick.
I'm sorry?
I had a question real quick.
Uh-huh.
For the infectious disease project, do we just email you through Canvas which disease and the microbe that causes it? Does it have to be anything longer than a couple sentences?
No, you just need to tell me what disease you're requesting.
Okay, thank you.
Okay. Make sure we finish chapter 1 and 10. I think we did, but just want to make sure. Emerging infectious diseases. All right. And let's go to chapter 2. And we started chapter 2?
We didn't start chapter
No.
2, we just finished chapter 1.
Okay. All right, thank you. Chapter 2, the chemical principles, the major goals, and a rough outline is shown in this slide. Know the terms. What's an organic compound? What's an inorganic compound? What's a hydrocarbon? What are polar and nonpolar molecules? What are hydrogen bonds? What's an acid, a base, and what is the pH? With respect to the four major biological molecules, be able to A, describe the relationships and the characteristics of monomers and polymers. Let me back step a bit and say the four major biological molecules. You can find three of them on every item you buy, like a cereal box. What are the four major biological molecules? What do you see on a cereal box? Nobody's going to take a guess. You should have had this in Biology 112 or 160, I guess. Um, the four major biological molecules are carbohydrates, proteins, lipids, and those three you can find on any item you buy, like a box of cereal. Although we don't use the term lipids, we'll use the term fats, which there are a little more lipids than just fats, but fats is the main one. And the fourth biological molecule, which you don't find in anything you buy, are nucleic acids. Describe the composition, structure, and function of these four biological molecules, and then be able to give examples of each. Any questions about what we're going to cover? All right. In this chapter, we're going to talk about the scale of things. Let me blow this up. Put that at the bottom over here. Oops, over there now. All right. So you should know the scale of things. The largest thing we'll observe or study would be something like a human. There are animals larger than a human. I think the great blue whale is the largest. And then there's something like an egg, chicken egg. That's actually the largest cell, the yolk of a chicken egg that all of you have seen. I once had a student who actually saw the yolk of a turkey egg. That's the largest cell you can see naturally on this continent, but the largest cell on the planet is actually the yolk of the ostrich egg, which I don't think any of you have seen that. The smallest thing you can see with your naked eye is about frog eggs. You can't see like a human egg cell. Eukaryotic cells are smaller than a frog egg, and those are something you need a light microscope to see. Below the size of a eukaryotic cell, we have prokaryotic cells. Below the size of a prokaryotic cell, we have a virus. Below the size of a virus, we have a large biological molecule, such as a protein or lipid or fat or carbohydrate, a very large carbohydrate. Below the size of a large molecule, we have small molecules like water. And below the size of a small molecule, we have atoms. There are things smaller than an atom, but in this course, we're not going to talk about them. Well, we'll not talk about most of them. I guess we do talk about a little bit protons and electrons, which are make up part of the atom. Being smaller than that, we don't talk about those in this class. You do need to only know the relative order of the size of things, meaning a person is bigger than a chicken egg, which is bigger than a frog egg, which is bigger than a eukaryotic cell, which is bigger than a prokaryotic cell. You do not need to know the exact sizes. There's a mistake in this graph here. I'll show you later when I show it to you in the next chapter. But they got 1 micrometer twice, once there and once there. That one there is the mistake. It should be 1 milliliter right there, not 1 micrometer. Any questions about any of that? All right, so let's get into chapter 2. What are organic compounds? Can anyone answer that?
Compounds that contain carbon.
Yes, most molecules that contain carbon are an organic compound, and all organic compounds contain carbon. There are some molecules which are inorganic, which do contain carbon, like carbon dioxide, but most molecules that contain carbon are an organic compound. Organic compounds are typically complex structurally, more complex than an inorganic compound, and they're held together mostly by chemical, covalent, chemical bonds, largely held together by the carbon, but some of them are not carbon. Most of them are carbon. And carbon is important in an organic compound because it can hold 4 other atoms, meaning 1 carbon molecule combined to 4 other atoms. And that's what usually holds the organic chemical compound together. An inorganic compound typically lacks carbon. Inorganic compounds are required by living organisms for the organism's growth, their repair, their maintenance, and their reproduction. Some examples of an inorganic compound are, of course, oxygen and carbon dioxide. And you know, carbon dioxide is a molecule containing carbon. And then maybe the most important example of an inorganic compound is water. Water is one of the most important and abundant molecules in living organisms. 70 to 80% of an organism is composed of water, at least the weight of an organism. It's made of water. Any questions about any of that? Sure, I've got everything I wanted to say about that. Yep. Here we're looking at the water, one of the important inorganic compounds. You'll note that water is 2 hydrogens chemically bound in a molecule, a covalent chemical bond to oxygen. But water is a polar molecule. It has a partial positive charge on the hydrogen, and this one too, and a partial negative charge on the oxygen. The reason why water is a polar molecule is because the electrons in this chemical bond right here are not shared equally between the oxygen and the hydrogen. The electrons are pulled and spend a little more time around oxygen than they do around hydrogen, making the oxygen a slightly negative charge and the hydrogen a slightly positive charge. I think your textbook calls it a slight charge, but I like to use the term a partial charge because it is not a complete charge. Like if the hydrogen were to give up the oxygen and give it to the OH group here, then the hydrogen would have a +1 charge and the OH group would have a -1 charge. The partial charge is not 1, it's something less than 1, but greater than 0. Any question about that? The polarity of water allows the formation of hydrogen bonds where in water there's an attraction between the partial positive hydrogen and the partial negative oxygen. That's an electromagnetic attraction, and that we call a hydrogen bond. In liquid water, this hydrogen bond breaks and reforms all the time. Let me see if I got a liquid. I don't know if you can see this. We're going to pour a little bit in here. You can see the water. Hopefully you can see that. The reason why the water changes shapes to the container is because it's a liquid. And it's changing its shape because that hydrogen bond is breaking and then reforming all the time. And that's why we call it a liquid. If the water were to have a permanent hydrogen bond where it would not break, the water would not change its shape to the container, and we'd call it ice. Ice is where the temperature of the water is such that the hydrogen bond does not break. And then if we were to permanently break all of the hydrogen bonds in liquid water, you'd have gaseous water where the water molecules have so much energy that they do not form a hydrogen bond. And without hydrogen bonds, you don't have liquid water, you have gaseous water. Any questions about any of that? Now, let me remind you that water has two chemical bonds. It has the hydrogen bonds between individual water molecules. And in liquid water, the hydrogen bond breaks and reforms all the time. In ice, the hydrogen bond does not break. And then in gaseous water, the hydrogen bond does not form. The molecule has too much energy and the hydrogen bond can't form. So that's one of the chemical bonds in water. The other is the polar covalent chemical bond inside the water molecule, holding the hydrogen to the oxygen. Any question about any of that? Well, let's see if this video will play. Oh, just a little slow. Oh, this isn't a very good one. That's right. Two hydrogen gas molecules, one oxygen molecule. And there we have two water molecules with the oxygen having a partial negative charge and the hydrogen a partial positive charge. And there's a hydrogen bond. You can see it's breaking and reforming all the time. Any questions about any of that? All right, let's move on. Not sure what happened there. Water is a good solvent, and that's because water can dissolve all polar molecules and ions. A polar molecule is a molecule like water where it has a partial positive end and a partial negative end. And polar molecules can dissolve in water and ions can also dissolve in water. Sodium chloride is a crystal that we call salt. And here we have the crystal sodium chloride, or the salt crystal, and it has a +1 sodium ion, the little green ones, and a -1 chloride ion, meaning the electron from the sodium moves to the chloride, giving the chloride a -1 charge and the sodium a +1 charge. That causes the electromagnetic attraction between the sodium and the chloride, and that's how we get a salt crystal. The +1 will be attracted to the -1, and you have all these ions coming together in a crystal formation. When you put this crystal in water, water is a partially charged molecule. The negative end of oxygen can come up to the +1 sodium in the crystal and then come around this molecule, which is kind of hard to show with that one. It's easier to show with the chloride ion, where the positive hydrogen can come up to the negative chloride ion and then surround the chloride ion and then pull it out of the crystal. And then the water will be like this, with the water molecules having the hydrogen, partial positive hydrogen, towards the -1 chloride ion. And then you have a number of these in the liquid water. And that will keep the chloride ion suspended in the liquid water. And the sodium ion as well, with the negative oxygen pointed towards the +1 sodium ion. And this we call salt water, where the chloride and the sodium are dissolved in the water. And that is how a ionic crystal can be dissolved in water. Any questions about any of that? So polar substances and ionic substances dissociate in water, forming a solute, and water is a solvent. It can dissolve some solutes, such as sodium chloride, in which case you no longer have the salt crystal, the sodium chloride crystal. You have the solution, which is the solute dissolved in the solvent. Any questions about any of that? So water is a good solvent. Polar molecules are hydrophilic. Hydrophilic means literally they love philic, water, hydro. That's the literal translation, but what we mean is that polar molecules can dissolve in water and they are attracted to water. Nonpolar molecules are repelled by water. We call it hydrophobic. Literally, this means fear of water, phobic, fear of water. So a nonpolar molecule does not dissolve in water. An example of a nonpolar molecule is oil. When you're making your salad dressing and you put the vinegar and the oil in the bottle, and then the vinegar and the water in the bottle, and obviously the seasoning. You then add the oil and you shake it up. And what happens to that salad dressing? The oil will separate from the water because the oil is hydrophobic. The oil is a nonpolar molecule and it does not dissolve in the water. And then you get the oil on top and the vinegar and the water and the seasoning below. The seasoning would be mostly hydrophilic, why it's in below the vinegar. Actually, the vinegar is mostly water, but the acetic acid in the vinegar, making the acid flavor of the vinegar, is a polar molecule, and so it'll dissolve in the water. Any question about any of that? That's not where I was. Where am I? Okay, hydrogen bonding formation between water molecules makes water a liquid molecule. The hydrogen bonding also makes water excellent at buffering the temperature. Those of you living in Western Washington or Western Oregon know that the western part of the state, the temperature is buffered compared to the eastern part of the state because of the Pacific Ocean. The water in the Pacific Ocean gives us our humidity and our rain, and the water buffers the temperature. While the temperature in Western Oregon and Western Washington is less hot in the summer than it is in Eastern Washington and Eastern Oregon. And then our winters are more mild. It's warmer in the western part of Washington and Oregon than in the eastern part of the state. And that's because water is excellent at buffering the temperature. Water can absorb some energy in the form of heat to break the hydrogen bonds. And so water can take some of that energy in their hydrogen bonds. And that means that the water molecules themselves will be taking less energy to lower the temperature. Meaning if you were to give, well, let's say 100 joules of energy to 1 cup of oil compared to 1 cup of water, liquid water, you'll find that the temperature will raise higher in the oil than in the liquid water. And the reason is because nearly all of the energy, we'll just say all of the energy, the 100 joules we're giving to the liquid cup of oil, all of that energy will be absorbed by the molecules of oil. So those molecules will move around faster and greater. The movement of the molecules is actually the temperature of the oil. And so the more those molecules move, the faster they move, the higher the temperature of the oil. When that same 100 joules goes to liquid water, some of that energy will be absorbed by the water molecules, and so they'll move around faster. That will raise the temperature of the water. But some of that energy, the 100 joules that we give to the liquid water, will be absorbed by the hydrogen bonds, and that will break the hydrogen bond. And therefore, the cup of water will raise less in temperature than the cup of oil. And then let's look at this cup of liquid water. The water molecules, which are the hottest, the ones that have absorbed the most energy, they're going to break all of their hydrogen bonds to the other water molecules. And then that water molecule will evaporate, leave the liquid. And that means the hottest water molecules are leaving the liquid water. And that will help cool or keep cool the liquid water because the water molecules with the most energy are the ones to evaporate. Any question about any of that? And that's actually why we sweat or perspire, and that is we're using liquid water to evaporate from our skin, and the hottest water molecules, the ones that absorb the most energy, are the ones to evaporate. And that takes the greatest amount of heat away with the water molecule evaporating. Any question about any of that? All right. On the planet Earth, water can actually split from H2O to H plus and OH minus. This H plus is actually a proton. The electron of the hydrogen goes to the OH minus group, and this we call the hydroxide ion, with the hydroxyl group with an extra electron having a -1 charge. That positive charge there is that's +1. The charged molecules participate in chemical reactions and actually give water its pH. Now, on the surface of the sun, there's so much energy hitting the water molecule that the water molecule, H2O, can actually break into 2 hydrogen ions and a oxygen ion. That does not happen on the planet Earth, at least under normal circumstances, because we don't have a high enough temperature to break water apart on the planet Earth. On the surface of the sun, there's enough energy, high enough temperature to actually break water apart. But it doesn't totally break apart on the planet Earth unless you put electrons in the water and then you get electrolysis. And we're not really going to talk about electrolysis. So remember, on the planet Earth, water, H2O, only splits into hydrogen +1 and OH -1. And that gives rise to the pH. And we'll talk a little bit about that in the next few slides. Any question about any of that? All right, the pH gives rise to the acidity or alkalinity. Neutral water, when H2O breaks apart into H plus and OH minus, will have equal numbers of OH minus and H plus. So the hydrogen ion concentration equals the OH minus concentration, and that is pH 7, pure water. Urine has a little bit more hydrogen ions than OH minus ions, then it's pH 6. Tomato juice, even more hydrogen ions than OH minus, and that would be pH 4. Lemon juice, pH 2, even more hydrogen ions in it. And that's about the most acidic thing that we eat, lemon juice, because you can't really drink it, but you could if you really wanted to, I guess. Stomach acid is the strongest thing that you're familiar with, except for maybe the acid in your car battery. That's actually around pH 1. Now, if the OH minus is higher than more concentrated than the H plus ions, we have increasing alkalinity or basicness. And seawater is a little more OH minus than H plus from the dissolved minerals in the seawater. And it's a little above pH 8. Milk of magnesia is a little above pH 10, having even more OH minus in solution than H plus. Also, the ammonia having a pH above 11. Also, bleach having a pH above pH 12, having even more OH minus groups than H plus. Oven cleaner is the strongest thing you've probably ever come across. It has a pH above pH 13, having even more OH minus groups than H plus. And then lime water is where we have the highest concentration of OH minus, and it's at pH 14. I don't think anyone's ever really come across lime water. So, increasing H plus concentration, we have increasing acidity, decreasing H plus concentration, increasing OH minus concentration, we have increasing alkalinity. And a base and an alkaline means the same thing, by the way. Any question about any of that? All right. So I mentioned that when water breaks apart into OH minus and H plus, it gives rise to the pH. And let me say exactly what the pH is. The pH is the amount of hydrogen ions in solution. Specifically, the pH is equal to the concentration of the negative log of the hydrogen ion concentration. So you can simply measure the hydrogen ion concentration, put it on a logarithmic scale, take a negative of that, and that's the pH. Any question about any of that? So the pH scale ranges from 0 to 14, increasing hydrogen ion concentration, increasing the acidity that will lower the pH from pH 7, and you can lower it as far as pH 0. At pH 0, just to tell you what pH 0 is, all of the water molecules have separated into H plus and OH minus, and the OH minus have been removed from the water at pH 0. So all you have left is the H plus. Decreasing the hydrogen ion concentration, increasing the alkalinity, raising the pH from 7. You can raise it all the way up to pH 14. At pH 14, all of the water molecules have split into H plus and OH minus. At pH 14, all of the hydrogen ions have been dissolved by something like in solution. So all the hydrogen ions are removed from the solution, and all you have left are the OH minus groups. Most organisms grow best around pH 7, so we can say they grow best from something like around pH 6 to pH 8. And if you go outside of that pH range, many organisms cannot grow. Like most bacteria grow between pH 6 and 8. And if you go at a pH range outside of that, then the bacteria cannot grow, cannot reproduce. Organisms need to maintain a fairly constant balance of cellular acids and bases to remain healthy. And hopefully you learn that term. We're not going to use it in microbiology, but the term for maintaining your acids and your bases is called homeostasis. If you've ever seen a kid chug a bottle of soda, soda is acid. And the reason why the child can maintain their health is because they maintain their balance of the acids and bases and homeostasis. Otherwise, they chug the Coca-Cola, it would kill them. But they have buffers in their body to maintain the balance of the acids and the bases. We won't talk about homeostasis, but it is important for an organism. Any question about any of that? See if there's anything else I need to talk about. Nope. All right, so we've talked a little bit about acids and bases. An acid is a substance that disassociates, giving up one or more hydrogen ions in solution. Like HCl, you put it in water, it'll dissociate into H plus and chloride minus, shown here, and that will increase the hydrogen ions in solution, increasing the acidity and lowering the pH. That's why HCl is an acid. A base, your textbook tells you, is a substance that in water disassociates, giving up an OH minus group. Like sodium hydroxide is a base. You put that in liquid water, it'll dissolve into sodium plus 1 and OH minus. That will increase the OH minus concentration in the liquid water, which will lower let's see, that will actually raise the pH. Increasing the alkalinity. I'm going to tell you that a base is actually a substance that either gives up OH minus or a substance that absorbs hydrogen plus. So you're getting this from me. Your textbook doesn't state it. I'm not sure why your textbook doesn't state it. So if we take like baking soda, sodium bicarbonate, there's the bicarbonate part. This bicarbonate can absorb H plus to form carbonic acid, H2CO3, and that will remove the H plus from solution, which will make the solution more alkaline. So sodium bicarbonate is a base, and it doesn't put OH in the solution. It removes hydrogen plus ions from solution. Any question about any of that? All right. So let's talk a little bit about salts and electrolytes. A salt is a substance that dissociates into a cation, that's a molecule with a positive charge, and an anion is a molecule with a negative charge. We're talking about a +1 and a -1, or a +2 and a -2, or some complete number, meaning it's not a partial charge, it's a +1 or a -1 or +2 and -2. However, a salt is a substance that neither cations or anions or H plus and OH minus. So water, which can break into H plus and OH minus, is not considered a salt. But sodium chloride is considered a salt because it can dissociate in sodium +1 and chloride -1. And obviously, sodium chloride is table salt. We actually do call it a salt. Now remember, though, that a salt is any substance that dissociates into a cation and an anion, like magnesium chloride breaks into magnesium +2 and 2 chloride -1s. So magnesium chloride in chemistry is also called the salt. Any question about any of that? An electrolyte is a substance that allows electricity to conduct in the solvent, like water. So sodium chloride, when you dissolve it in water, that will be an electrolyte because it'll allow the salt water to conduct electricity much better than pure water would. And that's why when you're swimming in fresh water, like a pond or a river, and there's a lightning storm going on, you're probably going to be safe because even if the lightning were to hit the water, you're not likely to get a charge unless you're really close to the lightning strike, because water, pure water, does not conduct electricity, at least very well. But if you're in ocean water or salt water, you should get out of that water immediately, because when the lightning strikes the water, it will conduct the electricity very well. And even if it's far away, you will still get the electricity from the lightning. Any question about any of that? All right, let's go blow that up. Let's talk about organic chemical compounds. We're looking at some of them here. There we go. An organic chemical compound tends to be complex. It tends to be a molecule. Let's look down here. It tends to be a molecule held together mostly by the bonds between carbon. And that's true for most of these. bonds here. All of these bonds are molecules held together from carbon to carbon to carbon, with the exception of right here and right there, and obviously right there, where oxygen is holding the molecule together. So most of the chemical bonds in an organic chemical, the chemical bonds are held together by carbon. There are a few where there's another molecule holding it together. Like in acetic acid, there's a chemical bond holding this hydrogen together through the oxygen. Some organic compounds can be simple, like acetic acid, that's fairly simple. Ethane, methane, they're simple. But some of them are pretty complex, like this phospholipid. Inorganic compounds tend to be all simple. Any question about any of that? So organic compounds always have, let's put that at the top there, always have carbon and hydrogen. So you look here, that's all these molecules are actually. And this one is mostly carbon and hydrogen. Same with this one. So they always have carbon, they always have hydrogen. Sometimes organic compounds can have other molecules, especially oxygen, but they don't have to. An organic compound that does not have oxygen or other elements, we call them oh gosh, what the heck is that called? A hydrocarbon. So hydrocarbons only contain carbon and hydrogen, and these are all hydrocarbons. Phospholipid is not a hydrocarbon because it contains other molecules like this phosphate group, and then there's an oxygen holding this together right there, as well as oxygen right there too. Do not memorize this chart, by the way. I'm just showing you some organic chemical compounds and hydrocarbons. So an organic chemical compound always contains carbon, they always contain hydrogen, but they may contain other elements, such as oxygen or nitrogen, sulfur, phosphorus. But organic chemical compounds are always held together by covalent chemical bonds. Compared to an organic chemical compound, trying to blow this up a little. Let's see where to blow that up. Oh, there it is. Compared to an inorganic molecule, organic molecules tend to be more complex and they're capable of more complicated biological functions. So, some examples of a organic chemical compound include ethane, propane, phospholipid, carbohydrates, lipids, fats, nucleic acids. Organic chemical compounds are always tend to be bound with carbon. Carbon is important because carbon can bind to far other atoms. Like in this organic compound, methanol, we have a central carbon binding 4 other atoms. Carbon can be found bonded together in a chain or a ring structure. Like here, this is a chain. The carbons holding the molecule together. Let's go back to here. I gotta blow this up. And this molecule here, it's largely carbon holding the molecule together. We do have an exception. There's oxygen holding it together here, here, and here. But all of these are stick structures. This is the ring structure. When we draw the ring structure, wherever we have a point, that is where a carbon is. If there is not a point in this ring structure, you have to literally put in the element. Like, for example, if this were to be oxygen right there, let's blow that up. It's not, but we'll use it right here. If this were to be oxygen there, you can't just draw the ring. You got to put the oxygen in there. And then the chemical covalent bonds drawn by the sticks there, or the line here, or two lines there. Any question about any of that? So the ring structure is these molecules here, and then that's the other structure called the chain. So there's a chain structure. The carbon skeleton is usually the backbone of the organic chemical compound. Like in this molecule, what's holding the molecule together is the bond between the carbon. So the carbon skeleton is usually the backbone of the organic chemical compound. And even for this molecule here, the backbone is mostly the carbon. It is true right there, and right there, and right there, that there's an oxygen holding the molecule together. But most of the molecules are held together by carbon. Organic compounds often have functional groups in them. These functional groups bind to a carbon, and the functional groups influence the chemical behavior of an organic chemical compound. Like methanol, the OH group is a functional group, and it gives the molecule different properties than methane. CH4, and this because methane has no functional groups. Methanol has one functional group, the OH, and this OH group gives this methanol different properties than the methane, the CH4. For example, methanol can dissolve in water because of this functional group. Methane cannot dissolve in water. Methane is a nonpolar molecule. Methanol is a polar molecule. Any question about any of that? Functional groups are important in organic chemical compounds because they can influence the behavior of the molecule and give the molecule new properties. So functional groups are responsible for most of the chemical and physical properties of a particular organic chemical compound, like a carbohydrate. A hydroxyl group is an alcoholic group. It's hydrophilic, and that's why methanol can dissolve in water, because methanol has an OH group. And that makes methanol a polar molecule. Methane is a nonpolar molecule, which cannot dissolve in water. Here we're looking at the functional groups. Let me blow this up. Shown in the red box, so you should know the alcohol functional group, found in lipids, carbohydrates, and nucleic acids, which isn't shown there. It's also found in proteins. Amino group, that's the NH2 group, found in proteins and amino acids. Why we call it an amino acid is because it has an amino group. The carboxyl group, also a functional group, CO with two carbon bond or two covalent chemical bonds, and then an OH group or COOH. Carboxyl group, found in organic acids, found in lipids, found in amino acids and proteins. And then the phosphate group, the last functional group we're going to talk about, found in ATP, DNA, RNA.
I have a question.
Uh-huh.
Carboxyl is the same as carboxylic acid, right?
No,
For the
carboxyl is a
functional
group.
group name.
It's a group, and a carboxylic acid contains a carboxyl group, but the carboxylic acid will have H2
Okay.
bound to this carbon here. And let me think, does that have, I think one H will be in this R position. I think the other H group binds to this oxygen, but I'm not positive of that. Okay?
Okay, thank you.
Okay. Now, frequently, an organic chemical compound can contain more than one functional group, such as found in an amino acid. All amino acids have an NH amino group and our carboxyl group, the COOH group. And so in an amino acid, they always have this central carbon and an amino group on one end and a carboxyl group on the other end. You should be familiar with all of the functional groups that I've shown in the red boxes. And why do you think I want you to be familiar with these functional groups?
Because they have different properties.
Yeah, they have different properties and they give the molecule a different property. But there are some other groups like the sulfhydryl group, the ester group, the ester group. I don't care if you know these. Why do I want you to know the carboxyl group, the phosphate group? Let's go to the previous slide. The OH group and the amino group. Take a look here. What's this molecule? What are these molecules? What are these molecules? They are the four major biological molecules, the molecules of life. These are the functional groups which are important in the molecules of life. The four major biological molecules we're going to talk about. The other groups, they're not so important in the four major biological molecules of life. So you do need to know the ones in the red boxes, because we're going to be talking about them, especially in this chapter. All right, so let's talk about an organic compound. Small organic molecules can combine into larger macromolecules. The small organic molecule, we call that a monomer. And then the macromolecule, made up of many monomers, we call polymers. And the macromolecules are made up of many polymers consisting of many small repeating molecules called the monomers. For example, starch is a large biological molecule. It is a macromolecule, and it is a polymer of many monomers. It is made up of many glucose molecules held together one after another in a what the heck is that called? A stick structure or a whatever that's called. Held together glucose one after the other. String structure, that's what I'm trying to say. So you just hook the glucose together on a long string, one after the other, and then you get starch. Any question about any of that? So of the biological molecules, 3 of them all have monomers, which make up the polymers. There is the 4th one, the lipids, which some of the lipids have monomers that make up some of the polymers, but there are some lipids which are not made up of monomers. But all the carbohydrates, the proteins, and the nucleic acids, they are made up of monomers, making the polymers. And to make the monomers a polymer, you take the monomers together, where the OH group of one monomer hooked together to the rest of the monomer, which we're just going to abbreviate with an R, can get together with another monomer, and there's an H group on this other part of the monomer, and the OH group comes off of this monomer, the hydrogen comes off of that monomer to make water, and then this R group binds directly to that R group. That we call dehydration synthesis, or a condensation reaction, where the monomers combine, making water. Well, OH comes off, the hydrogen comes off, and then the R group here, or the monomers, are linked together. And if you have more than 2 monomers, we call it a polymer. Synthesis reaction because you're making a larger molecule here than you're starting with. Dehydration because you're pulling off OH and hydrogen to make water. And then the reverse reaction can occur where you take the polymer, you add water, the water splits up here so that the hydrogen goes to one R group and the OH group of the water goes to the other R group. And then this polymer breaks into a monomer here and then another monomer there. With the hydrogen coming off of water going to this R group, the OH going to that R group. This is called hydrolysis, where you're splitting this molecule by adding water. And you should know what hydrolysis is if you've ever watched the movie Back to the Future. I think it's Back to the Future too, but I'm not sure what number it is, where Marty McFly says, "Ma, you sure know how to hydrate a pizza," where she takes this hockey puck-like structure, puts it into a hydrolysis machine, which adds the water and converts that hockey puck into this beautiful pizza that comes out of the hydrolysis machine. Any question about any of that? So we, in our cells, use dehydration synthesis to make larger molecules, and then we use hydrolysis to split the larger molecules to make the smaller molecules. Any question about any of that? So this is showing you again in a little better showing how you make a polymer. The monomer comes together with, this is already a small polymer, and the OH group comes off the monomer, the hydrogen comes off the small polymer. That monomer now binds to this small polymer, and that we call the polymer, and then water gets formed, and that's dehydration synthesis. Hydrolysis is the reverse, where water comes in, splits the polymer, the water breaks off, the OH group joins to the monomer, and then the hydrogen comes to this smaller polymer. Any question about any of that? So when you're eating a bunch of glucose or other sugar, you will undergo dehydration synthesis in your liver to make glycolate, I think. We don't make starch, we make glycolate in our liver. And then you
Glycogen?
can, glycogen?
Yeah.
Okay, glycogen. Yeah, I probably said it wrong. And then whenever your blood needs sugar, you will go under hydrolysis in your liver using water to break off the glucose from the glycogen and then release that glucose into the blood. All right, any question about dehydration synthesis and hydrolysis? All right, let's talk about the four biological molecules. We're going to start with the carbohydrates. Carbohydrates includes sugars, and we're talking about simple sugars, starch, other complex carbohydrates. Carbohydrates are an important energy source. So when you need energy, we frequently eat a carbohydrate, but we need carbohydrates for the structure of our body as well as our cells. And you need carbohydrates in nucleic acids as well. Carbohydrates consist of carbon, hydrogen, and oxygen, but not just any carbon, hydrogen, and oxygen. Carbohydrates have the general formula. For each one carbon, there are two hydrogens and one oxygen. And then in that carbohydrate, you just repeat that for n times. Like for glucose, n is equal to 6. And so the CH2O will be, for glucose, 6 carbons, 12 hydrogens, and then 6 oxygen. And so carbohydrates have this general formula. So you always have 2 hydrogens for 1 carbon and 1 oxygen. Simple sugars are the smallest carbohydrates, the monomers, and you can just make strings of these simple sugars linked together to make the larger carbohydrates, such as the polymers. You can classify carbohydrates into 3 groups depending on their size. The simplest sugars are the monosaccharides, the monomers. You can have 2 monomers hooked together to be a disaccharide, and sucrose is a disaccharide. And then there's the polysaccharides, which are many monomers hooked together. And then starch would be the best example of a polysaccharide, where you have many glucose hooked together in a string to make the starch. Any question about any of that? So let's look at the monosaccharides and the disaccharides. Monosaccharides are simple sugars. They always contain 3 to 7 carbon atoms, but usually simple sugars contain either 5 or 6 carbons. There's very few that actually are 3 or 7 or 3, 4, or 7. Most of the monosaccharides are 5 and 6. Sugars or monosaccharides are sweet and they're water soluble. A disaccharide is 2 simple sugars joined together in a dehydration synthesis. Let me blow this up. So here we have glucose, a simple sugar, joining together with fructose in a dehydration synthesis where water comes off this molecule and this molecule, and then that carbon there, this carbon here, binds directly to this oxygen there. So that carbon there is bound directly to this oxygen here to make the disaccharide sucrose, which we call table sugar. And then you can reverse that by adding water, which isn't shown here, but there it is there. And then you can add the water, break this molecule here, the hydrogen joins to the monomer here, which is fructose, and then the OH group joins to the other monomer, the glucose group here. And then this sucrose is split into fructose and glucose. Any question about any of that? So hydrolysis is just the reverse of dehydration synthesis, or vice versa. Oh, there we go. There's the water. Didn't see it over there. So you do add water to go that way. And there's the water. Here, was sort of hiding it with this. All right. You should know that the polysaccharides are the complex carbohydrates. Polysaccharides consist of many monomers. They can be straight or branch chains. Starch is mostly a straight chain of glucose hooked together one after the other in a linear chain to make the starch. Glycogen is similar to starch. It's the way animals store their glucose. The difference between glycogen and starch is the molecule is like this, but then it can branch where we have another chain going right here. And so this goes like that and then like that. Cellulose, another complex polysaccharide we'll talk about in a little bit. And chitin and dextran we're not really going to talk about, but they're all complex polysaccharides. Here we're looking at some starchy foods. Everybody knows that bread, cereals, pasta, rice, and potatoes are starchy foods. But did you know that beans and chestnuts are also have a large amount of starch in them? Beans, about a third of their weight is a carbohydrate, starch. And it is true they do have protein, so about a third of the bean's weight is protein, and about a third of the bean's weight is lipids. Chestnuts will be similar. I don't know the exact percentage of the weight, but the point is they both have a lot of starch in them. Any question about any of that? I suppose you could know that bread has some protein in it. Not as much as starch, but it does have some protein in it. The same. Oh, excuse me here. For some reason I wanted to sneeze. Ah. All right, let's move on. This table, not in the show. Let's try and blow this up a little bit. Shows you don't need to memorize this table. It's just showing you starch. It's largely hooked together, glucose hooked together to glucose in a linear chain. There can be a few branching chains in glucose, but most of them are linear. Glycogen is the same thing, glucose hooked together one after the other. And it's true the glycogen is a branch chain where the linear chain here branches there and there. And this one you notice branches there and there as well. Cellulose is what grass and many other plants have in them to make their cell wall, for example. And cellulose is simply glucose hooked together one after the other in a linear chain. And there you see the blue chain linking the glucose together, one after the other. And then the cellulose, we have one chain with this chain next to this chain, next to this chain. And you see these yellow links. These are hydrogen bonds linking this glucose to that glucose, this glucose to that glucose. And so cellulose has a whole bunch of linear chains of glucose. and they're all hooked together with hydrogen bonds to another glucose. That makes cellulose very difficult to digest, and that's why we humans cannot digest cellulose. It's just too complex of a structure for our stomach to digest it. Cows cannot digest cellulose either directly themselves, but they have microorganisms in their stomach. And if you know, cows have 4 stomachs, and the microorganisms can break down the cellulose and then break it down into glucose and then share that glucose with the cow. And that's why the cow can eat grass, because the cow with the microbes in its stomach can actually break down the cellulose. We'll talk about peptidoglycan later. Peptidoglycan is a carbohydrate. It's not really glucose, but it is monomers of simple sugar hooked together one after the other, and it's hooked together crosswise with the amino acids. And we'll talk about that later in a later chapter. But my point is that peptidoglycan does have some carbohydrate properties. It does have some amino acid properties as well. But peptidoglycan is important because it's found in the cell wall of bacteria. Any question about any of that? Is that a question or is that no question?
Sorry, no, I was going to ask you to zoom out on that picture for me.
I'm sorry?
I was just going to ask you if you could zoom out on it, but you did already.
Okay.
Thank you.
All right. Can I move forward?
Yes.
Okay. All right. How are we doing on the time here? We still have 30 more minutes. Let's talk about lipids. Lipids are a biological molecule, one of the 4 major, and they do consist of carbon, hydrogen, and oxygen, but most of the atoms are carbon and hydrogen. They do have some oxygen, but not very much. Lipids are mostly nonpolar and insoluble in water, meaning most lipids are hydrophobic. There is one exception, and that is the trying to remember the name of that, phospholipid. The phospholipid is more than just nonpolar and insoluble in water. The phospholipid actually has a polar part and is soluble in water. And that's the only lipid which is a lipid that is not nonpolar and insoluble in water, the phospholipid. All other lipids are nonpolar and insoluble in water. Lipids are structural and energy storage molecules. They play structural and energy storage molecule in cells and organisms. Lipids are a primary component of the cell membrane. And if ever you need a lot of energy, lipids contain the most energy per molecule of any other molecule. So let's say you're in Point Barrow, Alaska in the wintertime, and your car breaks down and you have a bunch of food in your car. And what should you carry if you have to walk 20 miles to Point Barrow on your feet in the wintertime? You want to carry lipids because as you eat it, you'll get more energy per pound of lipid or ounce of lipid than you would per any other ounce of any other molecule. Is that clear to everyone? I'm not saying eating lipids would be the best dietary thing. I'm talking about you'd get the most energy from eating lipids, like a fat. It may not be healthy because you get lots of fatty acids in your system and you could get high cholesterol and all that sort of stuff. But if you needed the energy, you want to carry the lipids because you'll get more energy per ounce than any other biological molecule. We break lipids into three types: simple lipids, complex lipids, and steroids. Let's talk about the simple lipids, a fatty acid. Fatty acids are a major component of many lipids, not all, but many lipids, such as fats, are made up of fatty acids. Fatty acids, let me blow this up, consist of a chain of carbons, shown right here. And it's mostly hydrocarbons, carbon and hydrogen. The fatty acid does have one carboxyl group, the COOH group right here, but only one in the entire fatty acid. We have two types of fatty acids, the saturated fatty acid and then the unsaturated fatty acid. The saturated fatty acids have a single carbon bond linking the molecule together. So this molecule here, let me blow that up. All of the carbons have a single carbon covalent bond linking the carbons together. So this is a saturated fatty acid. An unsaturated fatty acid has one or more double carbon bonds linking the carbon together. Let me end the show and go to the next slide to show you that. Blow it up a little. Right there, this is a unsaturated fatty acid. It has one double carbon bond. You can have one or more double carbon bonds in its unsaturated fatty acid. Wherever there's a double carbon bond, you put a kink in the fatty acid. And so instead of being a straight fatty acid, it has a kink in it where it bends where that double carbon bond is. Any question about any of that? Let's go back to make sure I've finished the last slide. Nope, that's it. All right. Simple lipids are simply a combination of a fatty acid and a glycerol molecule, such as a triglyceride, or what we call a fat, is three fatty acid groups linked to a glycerol molecule. And how you link that together in dehydration synthesis is you pull out the OH group from the fatty acid, the hydrogen group from the glycerol, and then you link that carbon directly to that oxygen in the glycerol, which is sort of shown right here. Pull that up. Where that carbon now links directly to that oxygen. As I stated, most fats in the body are a triglyceride, which has three fatty acids linked to a glycerol molecule. In a triglyceride, you can find these by just looking at your belly. Your belly tends to have fats that we call triglycerides. When you have a triglyceride that has one or more double carbon bonds, this is an unsaturated fat or an unsaturated triglyceride. If it were to contain only single carbon bonds, then it would be a unsaturated fat, which I don't have one to show you, but if you look at these two here and make this one the third there, that would be an unsaturated fatty acid, or excuse me, an unsaturated triglyceride. But you only need one double carbon bond in the triglyceride to make this an unsaturated fatty acid. And if you don't know, unsaturated fatty acids are better for your heart. They dissolve a little better in the bloodstream than a saturated fatty acid. And so these are better for your heart and for your having in your blood than a saturated fatty acid. Any question about any of them? So triglycerides are three fatty acids combined with one glycerol. They're the body's richest energy source, and they're stored as adipose in animals that we call fat. You just look down at your belly and you'll find triglycerides. Any question about the difference between a fatty acid and a triglyceride? A simple fatty acid and a complex lipid, the triglyceride. Another complex lipid is the phospholipid. The phospholipid is the main molecule in your cell membrane, also the main molecule in the bacteria cell membrane. Let me blow this slide up. Phospholipids are unusual in that they have a glycerol shown right here. And two of the positions of the glycerol are linked to a fatty acid. So there's a fatty acid there and a fatty acid there. The third group of the phospholipid does not have a fatty acid. It has a phosphate group attached to it. The R just stands for some organic chemical molecule there. This phosphate group is charged and polar. That means this head portion of the phospholipid is polar, and then the fatty acid portion is nonpolar. That makes phospholipids an unusual molecule because one portion of it is polar, the other portion is nonpolar, and this molecule can dissolve in water from the polar head, and it can dissolve in lipids like oil from the fatty acid portion. That makes this molecule biphasic, and there aren't too many molecules which are biphasic. You do know of two, meaning the phospholipid, and then soaps and detergents are also biphasic. And that's why soaps and detergents help us clean either our hands or our clothes. And that is the water can remove the polar molecule from our clothes, but water itself cannot remove the lipid molecule from our clothes. And you put in detergent, it will clean our clothes better because the detergent can get into the oil in our clothes. And then the other portion of the detergent can get into the water. And so the detergent can pull out the molecules, the lipids from our clothes, and then make it part of the dishwater to make the molecule dissolve in the detergent water of the washing machine. And that's why detergent help us clean our clothes, because it can help clean both the polar molecules, which you only need water to clean that, and then it can help remove the nonpolar molecules, such as oil in our clothes. Any question about any of that? All right. Phospholipids, when you put them together in water, they'll actually start forming a bilipid layer like this, where the heads will be facing the water here, and then the tails, which don't want to be around water, will come together, and they like being around nonpolar molecules, so they just come together like this. And then there'll be water on the outside here, where this head will be next to. And this actually is the main molecule of our cell membrane, which is a lipid bilayer, mainly containing phospholipid to make the bilayer of our cell membrane. Now, animal cells do not have a cell membrane, and so animal cells put cholesterol in their cell membrane. This is cholesterol right there. And the cholesterol helps strengthen and give support to the cell membrane. And if you wonder about eating cholesterol, you can only find cholesterol in animal tissue. You do not find it in plant tissue or in fungi like mushrooms because they don't use cholesterol in their cell membrane. So they don't make cholesterol. But animals do, and they put it in their cell membrane to help give support to the cell membrane. And they need that support because animals don't have a cell wall to help protect the cell membrane. Any question about any of that? All right. So when we put phospholipids together in water, they start forming this type of a structure. And then they can form this type of a structure where there'll be water on the inside and the heads will be facing the water. There'll be the fatty acid tails of the phospholipid on the inside. They like to be around nonpolar molecules, so they come together. And then on the outside, there will be the heads of the phospholipid, and it'll be facing the water. What does this structure remind you of? What does that look like? Anyone have a guess? What do you have in your blood cells? Not your blood cells. What do you have in your blood?
Plasma.
Red blood cell.
Red blood cells. Thank you. I wasn't sure what the first person said, so the second person answered it. This looks like a cell, and the red blood cells do have a cell membrane largely composed of phospholipids, where the heads will be facing the inside of the cell because the cytoplasm is mostly water. And then you have the lipid layer of the cell membrane being the phospholipid, which is a bilayer. And then the outside of that cell membrane, you have the heads, which also face outside the cell. And what is the blood mostly made up of? Water. And so these heads are facing the water. And so this is the natural shape of a cell, like a red blood cell. And phospholipids do form this structure when you put them together in water. They also form that structure, but we're not going to talk about it. Any questions about phospholipids? All right. So they are unusual for a lipid because they are both polar and nonpolar. They can dissolve in water. The head portion can dissolve in water, and they can dissolve in lipid. The fatty acid portion of the phospholipid can dissolve in oil, for example. All right, let's talk about another complex fatty acid, the steroids. The steroids are a little different, and that is they are not made up of monomer. I didn't ever mention it, but let's go back to this phospholipid. The monomers of the phospholipid are the phosphate group, the glycerol, and then the fatty acid. You put these together to make the polymer, which is the phospholipid. And then for the triglycerides, the monomers are the fatty acid, 3 of them, and then the glycerol, and then the polymer is the triglyceride. And these lipids are made of monomers. There's only a few of them. And the polymer is not very big. It's like on this one, it's only 4 molecules. On the phospholipid, it's about 1, 2, 3, 4 molecules. That's about the same. Didn't notice that until now. But there are lipids which are not made up of monomers. And these are the steroids and the sterols. These are one lipid that is contained by one molecule, a complex molecule. like cholesterol shown here. This is one molecule, which is a lipid. It's also a sterol. I'll talk about that. A steroid is four interconnected rigid carbon rings, characteristic of this group. And so A, B, C, and D are the four interconnected carbon rings making up a steroid. And usually there's some attachment, molecules attached to it, like this part here, and that there, and that there. And that's the steroid. If there's an OH group attached to a steroid, we call it a sterol. So cholesterol is a sterol because it has an OH group attached to the molecule. Any question about any of that? Steroids are mostly nonpolar. In fact, a steroid is totally a nonpolar. The sterol is mostly nonpolar, but it does have one polar region, the OH group. That is polar, this portion right here. Cholesterol can be in your blood, and it doesn't dissolve well in your blood because it's not very polar. It's mostly nonpolar. And cholesterol usually is in your blood around other molecules. I don't remember what it is. It's some either a a lipoprotein helps dissolve the cholesterol in the blood, or some other molecule is in the blood, which helps dissolve the cholesterol and suspend it in the blood. Cholesterol does not suspend well in the blood, and that's why the trouble with cholesterol is it can tend to lead to clogging of your artery, especially when the cholesterol comes out of solution and then coats your capillary, especially the capillaries in the heart, causes problems. Any question about any of that? Cholesterol is only found in animal tissue, and it's mainly because cholesterol is put in the cell membrane of animal cells to strengthen and support the cell membrane. So if you want to avoid cholesterol in your diet, you simply need to avoid eating animal tissue. And then if you eat very lean beef or other meat, you get very little cholesterol because cholesterol is largely in the fat of animal cells, animal tissue. So if you eat lean meat, you get less cholesterol than if you eat fatty meat. Any question about any of that? Remember, steroids and sterols are a lipid, but they are different in that they do not have any monomers. They are simply a macromolecule. I can't call them a polymer because they don't have any monomers making them up. All right, let's briefly talk about proteins. Proteins are the third of the biological molecules. They are essential in cell structure and function. Proteins may be structural proteins found in bone, muscle, and hair. Proteins may be enzymes which speed up chemical reactions. Proteins can be transport proteins, which help move chemicals across the cell membrane. Proteins can be regulatory proteins, which regulate body functions, like a hormone. Like a child produces growth hormone, which helps the child grow. Proteins can be responsible for movement. The reason why my hand is moving is because of the proteins in my arm, which are contracting to make the muscles move. In bacteria, if the cell can move, it is because of a protein, which is flagellin, which causes the flagella to beat. So all movement is by protein, both an animal like the walking or the flying for a bird, or the swimming of a bacteria from the flagella moving. Proteins are also responsible for some bacterial toxins. So if a bacteria produces like an exotoxin, that's likely to be a protein. And then proteins can also be antibodies. These are proteins our body makes to help defend us by binding to a foreign invader, like a bacteria causing an infection in your body. We can make antibodies, which then bind to that bacteria cell, which is invading your body. This tells you that proteins have a lot of different functions in an organism, and proteins have more roles and more functions than any of the other biological molecules because they differ more than any other biological molecule. They differ more than carbohydrates, they differ more than lipids, they also differ more than nucleic acids. Any question about any of that? All right, the reason why proteins have more function and differ more than all the other biological molecules is because proteins are made up of monomers, the same as like a carbohydrate. The complex carbohydrate is made up of monomers, but carbohydrates only have a few monomers between, I don't know, a handful to maybe a dozen monomers, depending on which monomers you're talking about. And for most of the monomers in the human body, there's only really like a handful of them, glucose, fructose. Most of the other simple carbohydrates will be converted into glucose. Proteins, on the other hand, are made up of monomers that we call amino acids, but there are 20 different amino acids in humans, and most cells have about 20 different amino acids. If a cell has a different amino acid than we do, they usually throw out one amino acid and then replace it with another. In some bacteria, the highest number of amino acids found in the living organism is only 23, which is pretty close to 20. So even though they have different amino acids, they throw out other amino acids. So most cells have about 20 amino acids in them. Amino acid is shown here. Let me blow this up. It has a central carbon, which everything binds to, and one end it has a hydrogen on it. The other end has an amino group on it, and then the other end, opposite of the amino group, has the carboxyl group on it. And then the third group, or the fourth group, I guess I should say, has an R group. This R group can differ. There are, in humans, 20 different R groups. And this psi group, the R group, is what makes the amino acid which amino acid it is. So you have one different R group for each different amino acid. So humans have 20 different R groups because we have 20 different amino acids. This is the generalized shape of an amino acid. You should know it. Tyrosine has this R group on it. And you simply link these different amino acids together to form a protein. Many amino acids linked together form the protein. Here we're showing you some of the 20 different amino acids. You do not need to know the different amino acids, so don't memorize that. But the simplest amino acid is glycine, where you simply have in this R group an H. Alanine has a CH3 group. Valine, 3 CH groups. Leucine, 4 CH groups. And then isoleucine, also 4 CH groups. All of these R groups are nonpolar. But our previous one, where was it? I lost the show for some reason. tyrosine, this OH group is polar. This group, or this part of the molecule, is nonpolar. This is a nonpolar R group. This is I already talked about tyrosine. Histidine is, this portion is nonpolar, and that portion is nonpolar. But this group and that group of the molecule is actually polar. On this one, it's mostly nonpolar, but this group is polar. And then that's a nonpolar amino acid. So in amino acids, all amino acids have polar groups. The amino and the carboxyl group are always polar. And some of the R groups have polar regions to them. And then some of the R groups are nonpolar. That makes a protein have both polar regions and nonpolar regions. If you have a molecule that is in water, this protein will have the polar regions of the protein on the outside of the molecule, and then the nonpolar regions, say the nonpolar R groups, facing the inside of the protein, so that the nonpolar groups are away from the water. And then again, if you have a protein in a lipid, like a protein in the cell membrane, for example, the nonpolar R groups will be on the outside of the protein, and then the polar regions of the protein will be tucked away on the inside of the protein, so that the nonpolar regions are away from the lipid, and then the polar regions of the protein are on the outside facing the lipids. Any question about any of that? So proteins are another molecule which are biphasic, and that is proteins have both polar and nonpolar regions. All right, any question about any of that? I think it's time to end the class. Let me check on that. Sorry, I don't remember the exact schedule of the summer, but I think we end at 6:50. Yep. All
Yeah,
right, any
yeah.
questions? If there's no questions, I will see you in the lab at whatever the lab time is. I think it's 7, but I'll have to look. All right. Bye.